Hello guys, welcome to my channel. In today's video we are going to study about the cell membrane. So as we are studying about the cell, so the first thing that we need to understand is the cell membrane. Right, so a living cell is a self-reproducing system of molecules held inside a container. So the cell, what's a container? The cell is a container. So that is a living cell. It's a self-reproducing system or a factory of molecules. So that container, that cell, the wall of the cell is called as a plasma membrane or a cell membrane, or you can call it plasma membrane. What it usually build of? It's built of a protein studded fatty acid film so thin that it cannot be seen directly in the light microscope. So the membrane is really thin that you cannot directly see in the light microscope. The structure of plasma membrane is simple. So it consists of a two ply sheets of lipid molecules, or you can say it's a lipid bilayer. So the plasma membrane is a lipid bilayer, and the thickness of it is about 5 nanometers, or thick up to 50 atoms. Right, so this much its thickness, into which the lipid bilayer, it's not only the lipid bilayer the membrane is, because it also has a protein inserted in between the lipid bilayer. Right, so the what's the function of plasma membrane? What's the function of the cell membrane? So it's like, if you see, like, suppose this is a cell, and like this is a cell inside, and this is a cell membrane. So like, and there are like organelles and molecules inside the cell. So the basic thing, like the cell membrane protects the inside function to stop going outside and the outside going inside. Like it, the inside molecule will not be mixing with the outside environment. But like, beside this is just a general thing, it does more than that, because the cell membrane, so it does more than that. So if a cell need to survive and grow, so it need nutrients, and so that nutrients must, like if it's an outside environment, so like the nutrients should go inside for the growth of cell and reproduction of the cell. So, and like there, if there is any waste inside the cell, it should go outside. So the plasma membrane is also doing that thing. So it has a highly selective channels and transporters. So like proteins that allow specific ions and small molecules to go, to be imported and exported. So this is some of the functions of the plasma membrane. So what the plasma membranes do? So like, as we discussed, like it export and import the small molecules and ion. So I'll just write down here, export and import molecules and ions. And then the other function is it also receives information from the outside environment. It also receives information of outside environment, that if there is any changes in the environment, so then how the cells should respond to them in the appropriate ways. And also uh, it has, suppose if the cell enlarges, so the cell membrane also enlarges. Like it, the cell membrane never tear off or never be like loses continuity, but it adjusts as per the size of the cell if it's enlarged in the area. Right, and if the membrane is like pierced, if membrane is pierced by anything, it neither collapses like a balloon nor remains torn. Instead it like quickly reseals. So these are some of the main features of the cell membrane. Okay, right. So and so the simplest bacteria having only a single membrane, so that is the plasma membranes. Whereas eukaryotic cells, if you don't know what are eukaryotic cells, so eukaryotic cells means that have plasma membrane, and also there it has an internal organelles as well. So that is like Golgi apparatus, reticulum, endoplasmic reticulum, mitochondria, and they also have their own membrane too. So that's the difference. Also contain internal membranes that enclose intracellular compartments as well. So that is the intercellular compartments are Golgi apparatus or mitochondria or otherwise. Although these internal membranes are considered, so the plasma membrane and the internal membrane for the eukaryotic cells are constructed on the same principle as the plasma membrane. But there are just subtle differences in their composition, especially in their resident proteins. And regardless of their location, all cell membranes are composed of lipids and proteins and share a common general structure. Right? So, as we discussed, like the plasma membrane is lipid bilayer. So, how it is arranged? So, I'll just draw a simple diagram. All right, let me do it here. All right, so this is just a simple diagram, and like, so this part is the lipid molecules. All right, so this part is a lipid molecule, and this is, for example, that's a protein inserted in between the lipid bilayer. All right, so this is just a section of plasma membrane or the cell membrane. So this is how the lipid bilayer. So these lipids in the cell membranes combine in two different, very different properties in a single molecule. So each lipid has a, like, this is suppose a one lipid. So if you remember, we studied in fatty acids. So like the fatty acid molecule has, some of the fatty acid molecules are amphipathic. So that means it has an hydrophobic and an hydrophilic end. So that same kind is used in the cell membrane as well. That each lipid has an hydrophilic head, and hydrophobic tail. All right, so as we studied in the fatty acids, so this is used in the cell membrane as well. And the most abundant lipids in the cell membrane are the phospholipids. So which lipids are most abundant in the cell membrane? They are phospholipids. All right, the most abundant and the most common phospholipid in the cell membrane. You have to remember this. So the most common phospholipid in the cell membrane are, I'll write it down here. It is phosphatidylcholine. So these are the most common type of phospholipid found in the cell membrane, right? So phosphatidylcholine. So the structure of phosphatidylcholines would be like this, something like this. So for example, this is choline. Okay, I'll write it down here. I don't think you will see, but like for example, this is, okay, choline. Then this is attached to a phosphate. All right, phosphate, and phosphate is attached to glycerol. If you remember in the fatty acid we studied glycerol. All right, and the glycerol in turn is attached to two tails of hydrocarbon tails. So these are that are hydrocarbon tails. Right, so this is the structure of phosphatidylcholine. That is the most common type of phospholipid found in the cell membrane. And if you want to see the chemical formula, or like a chemical structure rather, I will post the picture here so you can see how it has the choline, then the phosphate and the glycerol group, and it also has that hydrocarbon tail. And especially if you see, the second tail has a double bond as well. So with the phosphatidyl choline, so there is another major lipid also found in the cell as well. This is the most common one though, but beside this there is one more that is found in the cell. So that is phosphatidyl ethanolamine. Ethanolamine. So these two major lipids are found in the cell membrane. So that is phosphatidylcholine and phosphatidylethanolamine. And this lipid bilayer range actually is 5 to 7.5 nanometers. Something like that, the thickness of that cell membrane. Right, so beside the phospholipid, there are glycolipids as well present in the cell. Okay, so I'll write it down. Glycolipids too, and the glycolipids the one of the glycolipids is uh, galactocerebroside. Galactose, that's the one of the kind present in the cell membrane. And all the structures you can see in the image here. So beside like this phosphatidylcholine or the phosphatidylethanolamine, there is one more structure is phosphatidylserine. That is also one of the kind of phospholipid, and you can see the glycolipid structure as well on your screen. Right, so these are the different types of membrane lipids that are all amphipathic. Right, so the hydrophilic molecules dissolve readily in water because they contain either charged groups or uncharged polar groups that can form either electrostatic attractions or hydrogen bonds. So we'll see how it does like that. I will this. Okay. All right. So as I mentioned, so let's take an example. So for example, this is water, hydrogen, oxygen. But I don't have different colors, but so this is okay, I'll write it down. This is water molecule, and let's take an example of an hydrophilic molecule. That's an acetone. Let's take an acetone as an example. All right, so that will be like this. All right, so acetone. Okay, I'll write it down here. Acetone. So this is an hydrophilic molecule, acetone, and that's the water. So how hydrophilic molecule reacts when it goes into the water, right? So so this has a plus charge, this has a minus charge on it. So as the same as this has a plus charge and this has a minus charge. Okay, so suppose this goes inside the water. So what happened? So the water will form an electrostatic attraction and will also form an hydrogen bonds as well with the hydrophilic molecule. So for example, if this goes in the water, so this will form an electrostatic attraction and a hydrogen bond as well. So for example, this as if this molecule goes in the water, so this will join with this. Okay, we'll write it in the big one. So just to explain, and so that is, so this is hydrogen, that's an oxygen. So I cannot make like this, but I'm just making, and this is hydrogen. So the water is all around this, all right? So hydrogen, oxygen, hydrogen, and there will be hydrogen bonds again. All right. So it is surrounding, and here it will bond an electrostatic attraction with the oxygen. Hydrogen, then there will be hydrogen bonds. All right, so it will be like this. So this will be surrounded by water, and like it will be joining with the water, intermingled with the water with the electrostatic attraction and with the forming the hydrogen bonds in the water. So this is how hydrophilic reacts. And so the hydrophobic molecules are by contrast are insoluble in water, as we know that, because all or almost all of their atoms are uncharged and non-polar. Like the acetone molecule for the hydrophilic is charged and polar, but like for the hydrophobic they are uncharged and non-polar, so they therefore cannot form any favorable interaction with the water molecule. So how they will react with in the water? So the hydrophobic molecule, let's see, I will put it here, and let's, for example, the hydrophobic molecule is 2-methyl propane. Right, for example, the hydrophobic molecule is this 2-methyl propane. So as this does not have any charges and it's non-polar, so when this goes into the water, as this goes into the water, so the water molecule will form a cage around it. So what the water will do? So water will form a cage-like structure. Why the cage-like structure? So water, because the water need to mix in order to maximize their hydrogen bonds with each other. So as soon as this hydrophobic molecules goes into the water, so like water will try to make as more as hydrogen bonds inside the water. Okay. Right, so like this, it will just keep on bonding with each other, and then it will form a hydrogen bonds as possible as right. So it will form a cage-like structure to maximize their hydrogen bond with each other, and it will be surrounded by this. And because this cage-like structure is more highly ordered than the rest of the water, its formation requires free energy. So this energy cost is minimized when the hydrophobic molecules cluster together. That is the reason why when you see animal fat or oils found in the plant seeds coalesces into a single large drop when dispersed in water. All right, and the amphipathic molecules, that is such as, so because of this reason, so the hydrophilic head faces water on surface of the lipid bilayer, and it will connect with the water and it will form electrostatic attraction with the water, while the hydrophobic tails is always possible to join together or all shielded or like shielded from the water, and as they lie next to one another, like the filling in the sandwich, so it will be like this, and that's why it's always attracted towards each other, and this is how the bilipid layer is formed. So a lipid bilayer is formed. So the same forces that drive the amphipathic molecules to form bilayer help to make the bilayer self-sealing as well. So any tear in the sheet, so any tear in this cell membrane, for example, this is cell membrane. If any tear in this cell membrane will create a free edge that is exposed to water. And because this situation is energetically unfavorable, because it's requiring energy, the molecules of the bilipid layer will spontaneously rearrange to eliminate the free edge. And if the tear is small, this spontaneous rearrangement will exclude the water molecules and lead to repair of the bilayer, restoring the single continuous sheet. If the tear is large, the sheet may begin to fold in on itself and break into a separate closed vesicles. In either case, the overriding principle is that free edges are quickly eliminated. All right, so that was how it is hydrophilic and hydrophobic, and how it reacts and how it is arranged. So now the fluidity of a lipid bilayer depends on its composition. So the fluidity of a cell membrane, the ease with which its lipid molecules move within the plane of a bilipid layer, is important for the membrane function and has to be maintained within the certain limits. So just how fluid a lipid bilayer is at a given temperature depends on its phospholipid composition, and in particularly on the nature of hydrocarbon tails. So the closer and more regular the packing of tails, the more viscous and less fluid the bilayer will be. So if it's like really tight, it won't move. So two major properties of hydrocarbon tails affect how tight they pack in the bilayer is their length and the number of double bonds they contain. If you remember, if there is a double bond, it is really packed, and like because of the double bond, it gives a kinks to the hydrocarbon tails and it is hardly to move. So the shorter the chain length reduces the tendency of the hydrocarbon tails to interact with one another, and therefore increases the fluidity of the bilayer. The hydrocarbon tails of the membrane phospholipid vary in the length of like 14 and 24 carbon atoms, with 18 to 20 atoms being the most usual. So the tails will be like 14 to 24 and 18 to 20 carbon atoms are the most usual one in the hydrocarbon tail. So the most phospholipid contains one hydrocarbon tail that has one or more double bonds between the adjacent carbon atoms, and a second tail with just a single bond. So as we know, like it has the lipid bilayer. The one layer has two hydrocarbon tails. So one tail will have only the single bonds, but one tail will have a double bond as well. Alright, that is carbon adjacent carbon to carbon double bonds. So the chain that harbors a double bond does not contain the maximum number of hydrogen atoms that could in principle be attached to its carbon backbone. So it is thus said to be unsaturated with respect to hydrogen. So the hydrocarbon tail with no double bonds has a full complement of hydrogen atoms and is said to be saturated. So each double bond is unsaturated tails, each double bond in an unsaturated tails. So each double bond in an unsaturated tail creates a kink, a small kink in the tail, which makes it more difficult for the tails to pack against one another. For this reason, lipid bilayer that contain a large proportion of unsaturated hydrocarbon tails are more fluid than those with the lower proportion. All right. So that is the reason. So in bacteria and yeast cell, which have to adapt to varying temperature, both the lengths and the unsaturation of hydrocarbon tails in the bilayer are constantly adjusted to maintain the membrane relatively constant fluidity. For example, the cell makes lipid with the tails that are longer and contains a fewer double bonds. So a similar trick is used in the manufacture of margarine as well. So why we are discussing about the fluidity? Because the cell membrane fluidity is important for many reasons. It enables many membrane proteins to diffuse rapidly in the plane of the bilipid layer and to interact with one another, as is crucial, for example, in cell signaling. So and we'll discuss cell signaling later, but like that property is really important, that mem protein should diffuse rapidly in the plane of bilayer and it should interact with each other. And also it permits the membrane lipid and proteins to diffuse from site where they are inserted into the bilayer after the synthesis of other regions of the cell. It ensures that membrane molecules are distributed evenly between the daughter cell when a cell divides. That is also important for that the fluidity is really important in this. And under appropriate condition, it allows membrane to fuse with another and mix their molecules. If biological membranes were not fluid, it is hard to imagine how cells would live, grow, and reproduce. So that is why the fluidity is really important for the lipid bilayer of the cell membrane. We'll continue with the discussion of cell membrane again in the next video. Next, I think we'll see how the membrane assembly begins in the ER, endoplasmic reticulum, and so on. And we still have to study the internal integrated proteins in the bilipid layer and everything. But this is the some of the basic things of the cell membrane. All right, thank you guys for watching, and thank you for subscribing the channel. All right, thank you. See you in the next video.